Hi everyone. So in this lecture we are going to be looking at plant genetics, right? And this is going to set us up with a foundation from which we can then start talking about plant breeding and how we manage plant breeding. So to start, do any of you know what this is? Well, this is Hyacinthe, and that is the ancestral or wild relative to corn, right? It's what we think we developed corn from, or something similar to it. How about that? Any guesses? Well, that is the wild banana. And then that is the wild carrot. And that little thing there is the ancient peach, right? So, huge differences between our modern varieties and our wild or ancient relatives. And that brings us to the first kind of big idea of this lecture, first big idea of this course. And that is, we have modified the plant species that we cultivate from their wild relatives or ancestral state to better suit our needs, right? Pretty much every plant that we cultivate has been modified in some sense. And we've done that for much of our history through breeding, which we'll talk about. Right, so here's an example. Here's that peach again, right? So if you look at the ancient or the natural peach that would have been around in 4000 BC and compare it to our peach now, you see there are huge differences, right? One of which is that artificial peach is much bigger. It's about 64 times larger than the ancient variety, right? There's a greater or larger percentage of it is edible flesh. The skin is different, it's softer. It contains more water, it contains a little bit more sugar, and there's way more varieties, right? Ancient peaches, there was about 3 known varieties. They were only found in China, and now we have over 200 varieties, and they're found in various locations throughout the world. So huge differences between the ancient wild peach and the modern peach that we grow, right? And this is true for a whole host of crop species. And again, much of this throughout history has been done through breeding. And I'll talk about it real quickly here, but we'll get more into it in the next lecture. But breeding is simply selecting a female and male parent with unique and desirable traits and crossing them to produce offspring with both sets of desirable traits, right? So we have plant parents that have particular traits that we want. We want to pass those traits on, so we breed them together. And the idea is that the progeny or their offspring will also have those traits. And we just do this through over time and over time and over time. And ideally, if you're doing it correctly, you will have these traits either increase or become fixed in that population. So there are a number of these desirable traits that are common amongst all of our kind of plant species that we've domesticated, and that's essentially what we've done. And those are them listed there, right? So we've bred for gigantism. So we've typically bred for bigger plants that produce larger fruits or have larger roots in case of storage organs, right? If we're talking about that wild carrot that we saw there compared to our domesticated or crop carrots. We have bred for fewer seeds and the loss of seed dormancy. And we'll talk about seed dormancy coming up next week. But it's essentially this idea that, especially in our wild plants, our seeds are dormant for a certain period of time, and we have to satisfy these conditions to break that dormancy and allow the seeds to begin germinating. Well, that's kind of a hindrance if we want to keep producing new crops. So we've kind of bred that out or reduced that in a lot of our crop species. We have bred for our plant species to mature earlier. And we've also bred for a reduction of plant defenses. So whether those are physical defense, so thorns, or chemical defenses that they produce. And here's just a kind of a cool experiment to highlight the power of breeding, right? So here, this was done at the University of Illinois, and it's a selective corn breeding experiment that they did starting in 1896, and it ran until 2008. And what they were trying to do was increase the protein percentage or protein content in their corn kernels. And they started here with a variety. Can I get there it is. They started right here when it was about maybe looks like 11% protein, right? And then from that, they started breeding two different lines. And they bred a line to have a high protein percentage, and they bred a line for a low protein percentage. And you see, as they went through these generations and successive generations, you could see the result that it had, right? That high protein that they were breeding, it increased. The protein percentage of that corn increased all the way up until 2008, where now you're at the end of the experiment, their corn was about almost 30% protein, right? And the line that they were breeding for low protein got all the way down to around 5, right? So it really shows the power of selective breeding. And they also did some interesting things about halfway through. They tried to reverse it, and that's what you see going here. But really, this is just to, again, to highlight how effective breeding can be in altering plant traits. Now, to understand how that is working, we need to know a little bit about what determines a trait, right? So ultimately, what is determining a trait? A big portion of that is DNA. So DNA is the hereditary material of organisms, right? It carries the instructions for the development, functioning, growth, and reproduction of almost all organisms. I think there are maybe some bacteria potentially that are RNA based. Not positive. I'd have to look that up. Anyway, for most organisms, it's the hereditary material, right? And DNA, it consists of a sugar phosphate backbones, 2 of them that you see here, and then a sequence of these nucleic acid bases, right? And these bases join together to connect the backbones. And these nucleic acid bases, there are 4 of them: adenine, thymine, guanine, and cytosine. Adenine always binds with thymine, guanine always binds with cytosine. And it is the sequence of these bases that ultimately determines a gene, right? So a gene is a segment of DNA that codes for a protein, and it's this sequence of these nucleic acid bases that are going to determine what that protein is, and ultimately it's the interaction of that protein with other proteins as well as the environment that creates a trait. Right, and this is, you can see it unfold here, right? So we have a segment of DNA that consists of this unique combination of nucleic acid bases. Those bases get translated and transcribed into a chain of amino acids, which is a protein, and then those proteins interact with the environment and other proteins to create a trait. And in this case, that trait is green eyes. And this is highlighting the genotype and the phenotype, right? So the genotype is of an individual, that is the genetic makeup of an individual. That's the genes that an individual has, right? Whether it's a human or a plant. And then the phenotype is the actual observed characteristics. So we could have a genotype that codes for plant height. The actual observed height of that plant would be the phenotype. And that phenotype is influenced by your genes, or the organism's genes, as well as environmental factors, right? This is stuff you probably are aware of or you would have learned in other science courses. Now, more background information. Your DNA is packaged in what are called chromosomes, and it's just DNA that's all packaged together and wrapped up with proteins, essentially to allow it to fit in cells. Now, most organisms have 2 pairs of their chromosomes, right? And they refer to this as either 2N or diploid. So, for instance, humans have 23 unique chromosomes and then another copy of those 23 chromosomes. So we have in total 46, but there's only 23 unique ones, right? So you have 2 copies of each. And that's true of most organisms, although some species, particularly some plant species, can be triploid or tetraploid, so they can have 3 copies, 4 copies, 5 copies, 6, or more. I think some ferns can have up to like 200 copies, which is just crazy. And we refer to that, if you have more than 2 copies, as polyploidy. Right? And another important thing is that your organism's sex cells are haploid, right? This is because you get 1 copy from one parent and 1 copy from another parent, and then when they combine, you have your normal diploid organism that is going to develop. So here's an example of that. Because you have two copies of each chromosome, you have different forms of the same gene, right? So one on one chromosome, one on the other chromosome. And those pairs of chromosomes are called homologous chromosomes, right? And they have different versions, they could have different versions of the same gene. Right, so, and those different forms of the same gene are referred to as alleles. So here we have, this would be a pea plant, and we have a gene that is going to determine the height of the pea plant and a gene that is going to determine the coat of the pea seed, right? And here we see that there are two alleles. Why can't it? There it is. We have two alleles. In this case, when we're looking at the height of the plant, both of those alleles are the same, so they're coding for a tall plant. And here they are different. We have one allele on one chromosome that is going to code for a wrinkled pea seed, and the other that is going to code for a smooth coat of the pea. I guess it's pea fruit, not seed. Right? So, as I kind of mentioned there, your alleles can be the same or different. If they are the same, we refer to this as homozygous, right? So if we're looking at this hypothetical gene here, where both of those alleles are big B, that's a homozygous. It would be homozygous for that trait. Here, where they're both little b's, that's also homozygous, right? Both alleles are the same. If you have a situation where one allele is different from the other, we refer to that as heterozygous. Now, how does this affect the phenotype, right? The genotype, again, is the genetic makeup, so it would be the genes. It would, in this case, it would be the combination of alleles. How does that combination affect the phenotype, the ultimate trait that is going to be expressed? Well, typically, and it's not always the case, but there are dominant alleles and there are recessive alleles. Now, a dominant allele is going to be, the trait that it codes for is going to be expressed whether the plant is homozygous or heterozygous, right? So if you have a dominant allele in your genotype, it's going to express its phenotype that it's coding for. If you have a recessive allele, you need two recessive alleles. So you need homozygous recessive in your genotype in order for that trait that recessive allele is coding for to be expressed. And here we have an example. So now we're looking at pea flower color, and we see that the capital P is going to code for purple flowers. Well, if we have capital P, that's homozygous, we're going to get purple flowers. But also if we have these heterozygous genotypes where we have a capital P in each one, but the other allele is a lowercase p coding for white flowers, that purple flower, that capital P, is dominant. So the trait that is expressed is purple, right? Because that dominant allele is present, it's going to express its trait that it's coding for, which in this case is purple flowers. The only way you get white flowers is if you have two of these recessive alleles, and that's going to give you a white flower. All right, so that has given us a little bit of background. It's pretty introductory information on genetics, but kind of highlighted genes and alleles. The next step then is, how do these genes get passed down in successive generations, right? When our plants are reproducing, how are we getting the genes from this parent generation into the offspring? Well, our plants have two ways of reproducing. They can reproduce asexually or sexually. So asexual reproduction is vegetative reproduction, and that's what we're gonna end this whole course with later on. And asexual reproduction is essentially the plant is creating clones. Now, not all plants can be propagated asexually. Some have evolved structures to do it themselves, right? So some of those structures, I highlighted it in the intro lecture, right? Stolons in strawberries. They are these horizontal stems that grow, and from these stems we have new plants that can grow. So in this way, this strawberry plant can continue to propagate itself and expand where it's growing. Tubers, right? Like those are storage stems, like potatoes, right? We use those are a natural structure that is going to allow them to propagate, that plant to propagate vegetatively, right? So with asexual reproduction, it's important that the genotype is the same, right? That's important to remember. So the genes in your parent, in this case, are going to be, you're going to have the exact same genotype in the offspring or the plants that you propagate. And what this means is essentially you're going to have the same traits, the exact same traits as the parent. Maybe some variations if you're growing it in different environments, but for the most part, it will have the same traits. Now, sexual propagation or sexual reproduction in plants is the end result of that is the production of seeds, right? So sexual propagation begins with pollination, fertilization, and then we have your seed formation, right? So this is, if we're talking about propagation, this is the more common. It's the easier way to propagate. It's done more, like I said, more common in both the wild and in agriculture. And what sexual propagation or sexual reproduction does is that it mixes up the genes of the offspring. So whereas in asexual reproduction, your offspring are exact copies of the parents, this is not true in sexual reproduction. And because it's so important, I circled it just so you guys know, right? Because it is really important. This is going to affect how we manage seeds in breeding programs, how we are able to breed certain traits, and etc. Right? It's a really important aspect. Now, what is happening is in sexual reproduction is that you are combining gametes or sex cells, right? And you've all, I'm sure, have seen this in other science courses. But the process is that you are combining sperm and egg cells to create a zygote, and it's going to be a new organism that's going to grow. In the case of our plants, that is going to be a new plant that will grow and develop. So in our plants, the sperm cells are housed in pollen grains, and you see that here, right? That's the thing. Here's a kind of a diagram of a pollen grain, and within pollen, there's typically 2 sperm cells. And then the egg cells are housed in ovules, and that you can see them here, right? And this is the ovary of the plant. This is the stigma and the style. I don't. If you have plant science, we'll talk about that when we talk about pollination and fertilization. This is kind of just a brief overview. But within your ovary, you have your ovules, and here is a diagram, and that's your egg cell right there, right? So what's going to happen with sexual reproduction is that through pollination, so the transfer of pollen to the stigma up here of your pistil, and then there's going to be a pollen tube that grows, and ultimately that sperm cell from the pollen is going to make its way to the egg cell here. And from that, those ovules, the sperm and egg will combine. That's going to be your new embryo, and these ovules will mature into seeds. All right, so that's the process. But what's creating these sperm and egg cells is this process called meiosis. And in this case, what is happening is that, remember, we have 2 copies of our chromosomes, right? In all of our cells, same with our plants. In order to create these sex cells, we need cells that only have 1 copy, right? Because ultimately, the sperm is going to combine with the egg, and you're going to have 1 copy in the sperm cell, 1 copy in the egg cell. When they combine, you get your 2 copies, and you have your normal diploid organism, right? So this process of meiosis essentially breaks down these cells into versions that only have 1 copy of each chromosome and essentially 1 copy of each allele. What this means, and what I'm trying to get at, is through this process, through sexual reproduction, we have or can have a mixing up of genes, right? Because if we go back, right, we can have different genes on each of these chromosomes, and we'll have different alleles of these genes in these sex cells. And depending on what cells combine, you could have different gene combinations, right? And you can see that here. We have different combinations of different alleles of the father's genes and the mother's genes. And as a result, your children can have different combinations of these genes, right? And Again, if we want a formal definition of sexual reproduction, it's the production of new living organisms by combining genetic information from two individuals of different types. Now, the actual evolution of sexual reproduction is actually really interesting, right? If you think about life as a goal or purpose of life is to reproduce, there are a lot of costs associated with sexual reproduction, right? It's way easier to reproduce asexually. You can just grow a little bud of yourself, or you can have some sort of structure that allows you to reproduce asexually, have a little clone. It's way easier. Sexual reproduction, there's more involved in the whole process. And for our plants, they have to evolve flowers, they evolve nectaries. So there's energy that are going into producing the flowers. There's energy that's going into creating nectar to attract pollinators. There's a chance that there may not be other plants around in the area that they can pollinate with. There's all these extra costs involved in sexual reproduction. So if you are thinking from an evolutionary standpoint, why did this evolve in the first place? And the big reason it evolved is because it creates these new and novel gene combinations. And those new and novel gene combinations are really important when we are talking about defense against pathogens or parasites and diseases, right? And people have done studies about this. It's a leading theory of why sex evolved, and it's called the Red Queen hypothesis, if any of you guys are interested in. Essentially, sex is so important because it mixes up these genes, and that helps protect offspring from parasites and diseases. Because parasites and different diseases, viruses, bacteria, they have particular pathways that they use to access cells or tissues, particularly cells, right? There's certain pathways that they're going to use to access cells or body parts. And those pathways are dictated by like the organism's genes, right? So they'll have some protein that then that pathogen can recognize that protein, use it to get into your body, into your cells. If you mix up those genes, they may not have that same protein, and that pathogen may not have a pathway, right? And they've seen examples of this. So the good example that they give is there are snails I believe this is out in California, that exist in these small little ponds, and they can reproduce either sexually or asexually in the presence of no parasites of these snails. All the snails will reproduce asexually. But where there are parasites, they switch and they start reproducing sexually. And that is, again, to help protect their offspring from those parasites. So, little aside, but it's kind of neat to think about why this stuff evolved in the first place and the importance of it. And that kind of is going to lead into a little bit about the article that I'm having you guys read about. You know, we have bred those plants for those particular traits. And in the process of doing that, we have reduced the genetic diversity of our crop plants, right? We are getting rid of all this variation that you see here of all the organisms because we're breeding these plants to have we want them to have those particular traits. So we want those genes combinations to be the same. But that also leaves them vulnerable. And we'll talk a little bit more about that when we get into plant breeding. And the last thing that I want to mention is that, many traits are the result of the interaction of multiple genes, right? Tens or even hundreds of genes. So there are some that are, the result of few or one genes, like where we're talking about that pea flower color, right? And I have it just as one gene that's affecting it. But for many traits, it is this combination of all these genes that are interacting to produce a phenotypic trait. And we refer to these as quantitative traits. And some examples are crop yield, plant root architecture, the amount of milk produced by a cow. These are all the result of the interaction of tens or hundreds of genes that are working together to produce some sort of trait. All right, so I believe that is it for this lecture. In the next one, we will start talking more about plant breeding. We'll go into some Punnett square work to show how traits get passed on, and then we'll talk about how we can manage plant populations if we're trying to breed certain traits or prevent the infiltration of traits. So that's it, guys. Take care, and I will see you next lecture.